Good afternoon, everyone. My name is Dr. Courtney Callahan, a health scientist in the Center for Surveillance, Epidemiology, and Laboratory Services at the CDC. Welcome to the Laboratory Training and Services Branch webinar, The Interface of Biology and Chemistry, How Advanced Analytical Techniques Benefit the Diagnosis, Assessment of Treatment Efficacy, and Prevention of Toxin-Mediated Diseases. Today's webinar speaker is Dr. John Barr. Dr. John Barr received a PhD for the University of Virginia in chemistry. He is currently the chief of the clinical chemistry branch at CDC and supervises the development and maintenance of methods to diagnose, assess efficacy and medical treatments, and prevent toxin-mediated infectious disease, influenza, and selected chronic diseases. Since 1991, he filled critical needs within the CDC by establishing advanced mass spectrometry-based programs in several new areas, including anthrax, botulism, toxins, influenza, emergency response, counter-chemical terrorism, diabetes, coronary heart disease, and biomonitoring of persistent and non-persistent organic toxicants.
Okay, thank you very much, Courtney. I hope everybody can hear me all right. As Courtney mentioned, I'm going to be talking about kind of a new area of science, but it's really the interface of chemistry and biology and how the two really interact in order to generate more information that can be used to either diagnose disease or assess treatment efficacy and ultimately prevent this diseases. And as Courtney mentioned, today I'll be actually talking about our program for toxin-mediated diseases. I want to do a quick disclaimer. You're going to see some equipment in my presentation that we have in the laboratory, and any references to any commercial product, process, or services or company doesn't make for an endorsement by me or the U.S. government or the CDC. And also the findings and conclusions in this presentation are mine and mine alone, and they may or may not represent the views of the CDC. Okay, the objectives for this lecture are severalfold. First of all, I want to discuss some mainstream mass spectrometry methods for biological molecules, and this is going to be background information for the rest of the talk. Second of all, I want to present advanced methods for detection and differentiation of botulinum neurotoxins, and then we'll talk about methods that can detect and quantify anthrax toxins, and lastly, we'll be talking about what some of the toxin levels mean for clinical outcome. So just a little background on biological mass spectrometry. There's mass spectrometry is a fairly broad field, and there's many different techniques. The main two when you're looking at biomolecules are MALDI, which is matrix assisted laser desorption ionization, and that typically is coupled to a time of flight mass spectrometer. And the other one is actually combining liquid chromatography with electrospray ionization. That's what ESI is, and then doing some sort of tandem mass spectrometry on that. And so I'll show you just a few examples. This is how matrix-assisted laser desorption ionization works. You actually take a very small, as you see up in the right-hand corner, you actually can see a MALDI plate. Each one of those wells holds a microliter or less. And what you do is you take your solution that has your biomolecules in it, and you add it to a matrix, something like synapinic acid, and then they co-crystallize together. And so that's what you'll see down in the middle is what a co-crystal would look like in cartoon form. You then hit that with a laser, and the laser is tuned so the synapinic acid or the MALDI matrix will absorb energy, but the biomolecules really don't. That's what makes this such a soft technique. From there, then the matrix starts absorbing huge amounts of energy and finally explodes out. When that happens, you turn on 20,000 volts behind it, and anything that it is an ion will actually then go rifling into the MALDI-TOF. And um, uh, the way the MALDI-TOF actually works is since energy is equal to 1/2 mv squared, everything is given 20,000 volts of energy. And so as the mass change, the velocity changes by a square. And so basically small things fly faster than big things, and then we'll get the detector first. Now, because things always leave the source in kind of a spread of energy, there's something called an ion mirror, and this is what allows us to get to the high resolution that you see on modern day MALDIs. And so things that are having higher energy therefore go a little bit deeper into the ion mirror, therefore traveling a little bit further than things that they have lower energy. And so therefore, right before they hit the detector, they all line up in order to get a high resolution spectrum. Now, electrospray is slightly different. As I mentioned, that's being able to use liquid chromatography. And what happens is a solution that has your matrix in it flows down a narrow capillary. That needle then has around 5,000 volts, which really charges the droplets, either positively or negatively, depending on if you have a positive or a negative 5,000 volts on the needle. That then causes it to form a thin misting spray, which then can therefore be focused to go into the mass spectrometer. There's ion evaporation in between the needle and the inlet to the mass spectrometer, and so by the time it gets into the mass spectrometer, you've actually evaporated all the solvent molecules, and you have just ions going in. And so this is another very soft technique that can be used on fairly large molecules. Once they go in, there's different types of mass spectrometers that can be used. I'm going to actually, I showed you the MALDI-TOF or the time of flight already. The other two that you're going to see used in this talk is a triple quad and then a hybrid instrument. This is an example of a triple quad. As you can imagine, there are 3 different quadrupoles, and the first one you can select a mass. Then the collision goes into the second one, and then finally you can select a mass on your third one. So the collision in Q2 is actually fragmentation. And there's a lot of different ways you can do this experiment. One is you can do a full scan on Q1, and you can look at just one ion that would be of interest, like if you had a protein addict or something, and that protein addict stayed charged, then that would be called a parent ion scan. You can, if the fragment ion is a neutral, then you can scan Q1 and Q3 exactly with the difference in between, and that would be a constant neutral loss ion scan. What I'm showing here is actually a multiple reaction monitoring. This is the one we use the most because this is very specific, and it's probably one of the most sensitive methods in all of mass spectrometry. And that's when you do a targeted analysis. You know exactly what peptides or molecules you want to take a look at, and you know when they elute from the column. And so therefore, from before to after the retention time, you turn Q1 on and you park it at just the ion that you're interested in. You then do your collisions, and then Q3, you know what fragment ions it can do, and so you frag it on. You basically you park it at a specific mass, and then you can change that mass in order to look at more than one fragment. And as I say, this is an extremely sensitive and a very selective technique. Just an example to show you. This is actually a trivalent influenza vaccine that has all the different proteins in the influenza vaccine, and this is a tryptic digest, and as you can see, there's many peaks there when you do it in full scan. But by looking at things with MRM, this is what the MRM chromatogram looks like. And so when you're looking for H3, there's only that peak. H1, there's only that peak, and there's only the peptide for HB. You then can add internal standards in, which I have examples here. Those are things that behave just like the native molecule, but they're isotopically labeled with either C13 or N15, typically, and so they behave almost exactly like the native molecule, but they have a different mass, and so therefore they can be distinguished by the mass spectrometer. And so those are used to actually in quantification. The other thing is, since they're C13 and N15, they line right up with the unknowns here, and so you know the exact retention time that you are interested in. Okay, this is a hybrid mass spectrometer, and I'm going to show you a lot of data that we have actually gotten off of this instrument. This is a little bit more complex. So this uses an Orbitrap analyzer, which can give you very, very high resolutions. Not unit mass, but down to several decimal points out, and then very accurate masses. And so when you get the mass of a peptide, you can almost calculate, or you can calculate the elemental composition many times of that peptide. So that gives you confidence that you have the right parent ion, and then you do your MSMS. And the way it actually works is as you can see, you put all your ions in that are coming out at a certain retention time, and then they go into the Orbitrap. They start separating out in the Orbitrap very quickly, and then they go into, you choose just one of those ions, which then after it gets cooled goes into an ion trap, and you get MSMS. Well, that MSMS is going, you're picking at another ion that then can be cooled and go into the mass spectrometer. So therefore, you can get 20, 30 ions from very quickly as you're getting one high resolution spectrum. So you get an accurate mass parent ion, and you get LC-MSMS daughter ions, and this actually is very gives you high confidence when you sequence out a peptide or a small protein. And the way that it works is that basically peptides behave in a very predictable way. Where they typically, these are low energy collisions, so typically we see Y ions and B ions. B ions are where the charge remains on the N terminus, and then Y ions is when the charge resides on the C terminus. And basically, as you can imagine, if you had 1,000 molecules, you might have 2,000 that split at Y. I mean, 200 that split at Y1, and 500 that split at Y2, and another 200 that split at Y3. And because you get those populations, you get ions and peaks at each one of those sites, and then you can tell by the difference between those what the sequence is. And there's software that can help you do that and actually can predict what the sequences are from a spectrum. So that's just a little background on mass spectrometry. I thought I'd go in now into actually how we use this for infectious diseases, toxin-mediated infectious diseases, and we'll start with botulism. Botulism is actually one of the bigger challenges. It's the most toxic substance known to mankind, and therefore, at very, very low levels, it is public health emergency, and which means that you have to be extremely sensitive in order to pick up these tiny amounts of toxins in people's stool or blood or food in order to detect the toxin and confirm a diagnosis of botulism. There's basically 7 different serotypes, and they're called A through G. Human botulism is caused by only 4 of those, and there's A, B, E, and F. The other ones, C and D, cause botulism in animals and birds. G was found in soil, and there's no disease in any animal associated with it. But it's 150 kilodalton protein with a light and heavy chain. there's three main different types of botulism. There's foodborne, and that's one we think of when you improperly can food and um, uh, end up eating the toxin and getting sick. And there's been some dramatic outbreaks of that even recently. At a church supper um, in Ohio not that long ago. Many people got sick eating potato salad. Um, there's also infant and adult colonization, and that's actually when the Clostridium botulinum grows in the intestines and produces toxin and therefore making the patient sick. And there's wound, which we've been seeing quite a little bit of lately. This is typically associated with drug use like heroin, where there is Clostridium botulinum spores in the heroin, and when people inject it, it then grows in their wounds and can make them very ill. The traditional method for detecting botulism or botulinum neurotoxin was something called a mouse bioassay, and that's where you inject a mouse or pairs of mice with the sample, and then you wait 4 days to see if they develop symptoms and end up dying from the toxin. Uh, Also, the serotypes can be determined by this method by giving neutralization antibodies to different combinations of mice, and you see which ones survive and which ones die. The upside is it is extremely sensitive. The downside is that it takes 4 days to complete it and use a lot of mice, and it requires actually quite a large sample volume. So how does botulism work? Well, basically the toxin binds to coated pits on your neurons, then through endocytosis goes inside the neuron, where again there's a pH change, and then it cleaves proteins which are important in the snare complex, which are important for the docking of the synaptic vesicle and the release of neurotransmitter. That's why you go into flaccid paralysis. One thing I'd like to say is also that it really is a bad disease, because paralysis for some serotypes can last for months, and so basically the mind is still active, but the person is totally paralyzed and lasting for, as I say, weeks or months on a ventilator. So it is really a good disease to try to prevent. Each one of the different serotypes has a unique cleavage. I'll show you about F. F actually is a little unique. It has more than one cleavage depending on the subtype, but but uh, uh, they have different proteins and different sites on those proteins in which they cleave. So basically, we actually use that. And so in the body, you have a snare protein, it gets cleaved, leads to paralysis. So in the laboratory, we made peptide mimics. And there's several reasons that we went this way. Um, First of all, in order to get the specificity, we have monoclonal antibodies that we put on magnetic beads, and that is used to extract it. We then add the peptide mimics, and we look for the cleavage reactions by Maldi-Toff, which I talked about earlier. Um, and that gives you the serotype specific detection. So you know if botulinum is there or not. These monoclonal antibodies actually worked with Jim Marks at the University of California in San Francisco for a number of years, both where he was able to discover and then produce some extremely high affinity, very specific antibodies. And the mixture of antibodies that we use um, are for the heavy chain. We then go into um, the endopeptidase activity, which is in the light chain, and then that lets you know that you have a full toxin there. Um, the advantage of using the enzymatic activity is if you have one toxin, and if it can cleave peptides over and over and over. So basically, if it cleaves 1,000 times, that gives you 3 orders of magnitude increase in your sensitivity of the actual thing that you're detecting. And this enhancement or amplification of signal is really necessary in order to get these extremely low detection limits. So what we actually see is in the absence of toxin, you see just your parent ion, and in the presence of toxin, what you see is you see your product, your substrate ion go down, and you see the appearance of these product ions. And so that's kind of what the spectra look like. I'll show you a few examples. Um, this is actually one of the first outbreaks that we got to work on. This is from carrot juice, and so the top spectrum is from carrot juice. The bottom spectrum is from the serum of a patient that drank that carrot juice. And you can see these are really are are very easy to identify. And so moving on, um, this is an example of B from an infant serum, and as you can see, uh, you have your 2 peaks there, and this is F from adult colonization. This actually happens to be an F from a Clostridium botulinum, Clostridium baradii, and as you see your 2 peaks there, this one is much more sensitive than the other. And if we go on, this is an example from Alaska, which was a type E in seal fat. And the one below shows a type A in serum. We're actually quite pleased that we can now see wound botulism in serum, because this was always quite a challenge for the mouse bioassay because of the sensitivity that you need. So and this is a bivalent. So there are some strains of Clostridium botulinum that make more than one toxin, and it usually is a major toxin and a minor toxin. So this would be, as you can see, there's a type A on top, and that's a weaker spectrum. There's a type B on the bottom, and right here, and this is a much stronger spectrum. And so this one would be called a big B, little a type toxin. And so there's something else that classical techniques had real difficulty of being able to identify the dual producers. So right now, the detection limits of the mass spectrometry methods are about 0.05 mouse LD50. So in serum and stool, it's between 10 and 20 times more sensitive than the mouse bioassay, which is much more sensitive than the ELISAs. We actually are putting this right now onto simplified equipment that can go out into the LRN. On the type of equipment which can do also does bacterial ID by MALDI-TOF MS, and so right now we're just getting stuff together for FDA approval. Just to show you real quick, we've analyzed well over 1,000 clinical samples in parallel with the mouse. During that time, it was 98% concordance, and most of where we disagreed is because the EndoPep MS is more sensitive. Right now, we're not really running in parallel. We're just running samples. And the EndoPep MS assay has reduced mouse use at CC by more than 90%, which is a good thing. So. As I mentioned, that among the 7 different serotypes, there are also subtypes, and so like with A, I drew 5 of them out. There's 8 right now, but that are known, and then among these subtypes, there's something called toxin variants, and that's where there may be only a few different amino acids are different. The the thing is that all this diversity in toxin has made these a real analytical challenge, but there actually is an analytical opportunity here also, and that opportunity is when there are differences between the toxin subtypes and variants, those can be used to show commonality between outbreaks or differences between outbreaks, and so basically, first of all we do the antibody extraction, as I mentioned, and then we put it into the activity assay where we add our substrates. Once you remove those substrates, and that goes to the MALDI-TAW, we have the beads with the toxin still on it. With that, there's some opportunities that we can do, and that's analyze those beads further. Now, the subtyping method is less sensitive by quite a little bit, but if you get a nice strong activity assay, then you know that you can very likely get a subtype. And for that, as I mentioned, the toxin is still on the beads, so instead of just autoclaving them, we can do a tryptic digest where you get lysines and arginines. We use right now a combination of trypsin and chymotrypsin, and we use very rapid digest in a thermocycler at an elevated temperature, and then they go to MSMS on the type of instrument that I showed on the Orbitrap that I showed you previously. And the advantage is this is an example of 3 different outbreaks actually that we got in the same week, and 2 of them were in neighboring states, and then one of them was several states away. But this is when you start looking at these and you say, are these related? Are they separate? Should they be looking for national brands? Should they be looking in their cupboard at canning? And so it's good to know what the commonality is between these. And this one was actually quite dramatic. So 3 type A outbreaks. Now, if you look at the difference between a sequence of A1 and A2, the ones in orange are different between A1 and A2. And as you can see, there really are several different amino acids. So these can be, we can use LCM, SMS, and sequencing in order to exploit these differences and tell if you have an A1 or an A2 or an A3 or an A4 and so on. And I'm just going to pick just one peptide because one thing that we've been able to be very good at is getting extremely high sequence coverage where we get 80 to 90% of all these peptides recorded. And then, so therefore you just don't see one difference between them. You see often many peptides that are unique. But this is just one example of one peptide. And as you see, sample 1 on the bottom had a aspartic acid, and the one on the top had a glutamic acid. And basically, the way these go on a B ion is B1, 2, 3, 4, 5 is the E. So if you look at B5, they have a different mass, but if you look at B4, they'll have the same mass. And the same thing with the Y ions, you can count on the other side. And so this ends up being the one on the top, the second sample was an A1, and the one on the bottom was an A2. So we know that those outbreaks are not related because they have different types of subtypes. So we also then looked at 2 versus 3. Now both those ended up being A1s, but there is 2 amino acids difference between a variant, and it's a fairly common variant that we see in the U.S., and it's called an A1 silent B. It's because there's a gene for B that's incomplete, so it doesn't make the toxin. But there is a gene there for it. And this is just a couple different, and I'll show you this one peptide that we're able to identify. And as you see, the one on top is sample 2. The A1 that I showed you before is actually an A1 silent B, whereas the one down below ended up sequencing out to be an A1 Hall, and so therefore these three outbreaks going on simultaneously, they happen to be three different toxin types, either subtype or variant, and that tells you that they're probably not related. And so these are just some of the a short list of some of the subtypes that we have found over the last few years, looking in the U.S. Of different types of toxin. Some of the things that we learned from doing this work is, first of all, there is a F5 that does have a unique, and a type of F that does have a unique cleavage site. And so to validate our method, we had to look at every different type of subtype and variant that we get our hands on. And when we looked at the F5, sure enough, it actually cleave several amino acids down. I see my slide has been shifted, but where the two white ones, the LE is the F5 cleavage and the QK is every other single other F. The other thing is that when we look at F7, it needs 5 additional amino acids over on the N-terminus for the F7, which is the type of toxin from Clostridium baradii that actually needs to do that. And we've actually exploited that because F7 is much more likely to do a colonization or be in meat. And so you actually do get more clues at where you should look when you have a botulism case with an F7. So that's one lesson or a couple lessons that we learned, and these differences make sense when you look at the uniqueness of the ones. If you go across here, F5 is very different in the light chain from all the other ones, and then the next most different one is F7, which is about 60% different. If you look at the difference between serotypes, B and G have a light chain which is only 61% different. E and F is only 56. So F5 is actually more different than than some of these other serotypes are. The other thing is that we discovered, and I'll go through this fairly quickly, but we got a strain that actually had 3 different toxins associated, because before then only bivalents were known. This is the first trivalent one. This came from Chile, and originally it was thought to be a bivalent with A and F. But when we took a look at it, we actually got the cleavage for both F4 and F5 in it. And sure enough, we also did the LCM-SMS, and in white, you'll see this is from the A2. And then the next one, the F antibodies, actually pulled out F4 and F5, and then white is where we got the different sequences on both of those. And so we show those by both activity and by sequence. And this is the just one peptide out of many that we were able to do. It had both Y and B series. So the ratio that we got, we actually did do quantitation on these, and the ratio was 45 to 1 to 7.5, A2, F4, and F5. So this was a large A, little f, and the strain is called AF84. The original publication that thought it was a bivalent actually gave it 93 and 7%, which is close to what we get of 84 and 16. Especially figuring that with the F4 and the F5, they're going to react slightly different to F1 antibodies. So we were quite pleased to find a new trivalent strain. And just one more example. There was a strain discovered in California from an international infant botulism case, and it was thought to be a big B, little h, H being a new serotype. Um, when we at CDC were able to get a hold of this strain, within 1 day we showed the toxin could be captured by A antibodies, and we did that by doing a tryptophan digestion and showing that which type of antibodies actually could bind to the toxin. We also showed the enzymatic activity was the exact same as what we found in F5, suggesting that it is most likely an FA chimera. I'll just show that data real quick. The novel, this is on full-length VAMP. We saw that got cleaved. SNAP25 did not get cleaved. Syntaxin did not get cleaved, but VAMP did. And that VAMP cleavage site was at the exact same place as F5, where all other Fs are at another site. The DNA sequence matches very close to an F5. On that, it's I think it's over 90% in agreement, and there is the B. What's kind of interesting is because both the B and the F both cleave um, um, VAMP. We showed that when they were actually together, that you saw cleavage for B, you saw some that were cleavage for the F, and then you saw some that actually cleaved both of them, and a piece in the middle. Um, so then the next step was that the National Botulism Group down in EZID here at CDC actually did the mouse bioassay work and showed that H was indeed neutralized by A antibodies. We showed it, captured them, but they showed it actually neutralized them, and so it is an FA chimera, very much like there's other chimeras. C and D also make chimeras where you have a light chain for C, or similar to a light chain for C with a D heavy chain, and vice versa. So, um, but basically, just summary on the botulism, and I'll go fairly quickly. Basically, the methods are sensitive and they're rapid, and kind of the take-home message is advanced technology offers more sensitivity, faster detection, and more information when it comes to dealing with botulism. Moving on to Bacillus anthracis, and because I'm running a little late, I have to go through this faster. There's basically, among the virulence factor are two binary toxins, and they include, and there's several different types of anthrax. There's cutaneous, gastrointestinal, inhalation, and a newly formed injection. Inhalation is typically fatal, and it is often fatal even with antibiotics and high level of supportive care, as the 2001 anthrax letter attack actually showed us. One interesting thing, and I'll just point you there at the bottom, is that typically patients become rapidly ill, but then they often look like they're recovering, and then they relapse and they get very sick and die. And I think we're starting to understand this progression a little bit better. The three different toxins that makes are lethal factor, protective antigen, and edema factor. And protective antigen, the reason they do that is because if you get this in an immunization, it will protect you against anthrax. But the lethal factor and protective antigen bind together to form lethal toxin. Protective antigen and edema factor bind together to make edema toxin. And lethal toxin is a protease. It does MAP kinase cleavage. Edema toxin is makes cyclic AMP from ATP. And so the mode of action is thought to be that either in serum or on the cell surface, it binds PA, which then gets processed and you lose a 20 kilodalton portion to form PA63, which then can form a heptamer and octamer, which then combine either 3 or 4 edema factors or lethal factors, which then can get through endocytosis into the cells, and then from there, the either the edema factor or lethal factor make cyclic A and um, um, cyclic AMP or cleave MAP kinases. Um, Now we can actually separate these out. There are antibodies for lethal factor, and that will bind both free lethal factor and lethal toxin, the complex. There's edema factor antibodies that can do free edema factor or the complex, and there's protective antigen that can do free protective antigen or just the complexes. And that this way we can actually separate things out because of their enzymatic activity and their binding characteristics to get how much is free and how much is actually bound, or a total and a bound, and then you get free by subtraction. Um, and how these methods actually work is you bind them to monoclonal antibody, to magnetic beads, just like I showed you for bot. We add in the substrates just like for bot, and we go to Molly-Taw. The edema factor, again, we add in the substrate, and then we go to LCMS to do the cyclic AMP, and with the edema factor, edema toxin, and the lethal toxin, we could use the PA antibodies, then bind both, and then they go to the two different perspective methods, and you can get how much is actually the toxin. Just moving on, these are extraordinarily sensitive methods. Lethal factor is down at 0.0027 nanograms per mil. Edema factor at 0.00002 at 220 zeptomoles per mil. And then with protective antigen, because we don't have the enzymatic activity and we have to look at just the peptides, we're in the nanogram per mil range, which is still sensitive. So it's a combination of detection limit and the amount that's produced that can show you how early these can be detected. So we have some animal studies, and first of all, we found that the toxemia in both non-human primates and in rabbits is triphasic, where basically there's a rapid increase, a plateau, and then a rapid increase. So this corresponds fairly well to get sick, starts feeling better, and then relapses and gets sick and die. Now these are on log scale, so basically at the end when you see that going up, that actually goes up a lot because it goes up from basically from just a few nanograms per mil up to 1,000 nanograms per mil in some cases. This is the other thing we found that lethal factor is produced more than edema factor, and lethal toxin is higher than edema toxin. And basically in early phases of disease, you see a lot of free lethal factor and edema factor, but late in disease, you see almost all in the complex form. And we go on from there. And basically, both the edema factor and the lethal factor both do the triphasic. The other interesting thing is that between PA63 and PA83, basically throughout the infection, it's all in PA63 form. And I know I'm supposed to stop soon to get questions, but basically, the toxin levels seem to be very predictive of survival, and so those in red had died very quickly, where the ones in green lived out to be much longer, and this is probably because of the toxin level coming out of phase 1. Also, for those that got treated, those that stayed below a certain threshold, and it's always dose makes a poison, but those that stayed below a certain threshold basically all survived with antibiotic treatment, where those that went above a certain threshold didn't. And this is holding true also in some larger cyno studies. And if you look at the toxin in tissues, you basically you see that they're spread out among many different ones: lung, liver, kidney, lymph node, spleen, and that those that had died from multi-organ failure or sepsis, basically we had a spread among the organs, whereas those animals that died from meningitis had mainly the toxin in their brain. Um, some human clinical cases, and I'm almost finished here. Basically, from the animal studies, if you see it go up, then you know it's going to have a bad outcome, and if it continues, toxin level continue to fall, you know that they tend to survive. Um, Here's a an example of a human clinical case, and as you see that they continue to go down here, and then when they got AIGs, which is antitoxin, fell very quickly and recovered. This is the antibodies for the anti-PA antibodies. And you see they were low when they got AIGs. They jumped up here and then stayed high. For non-surviving people, we see the exact opposite. We'll see the antibodies fall, and we see the toxin do a U-turn and go back up. And so we have distinct patterns that really do indicate survival or not. This is those that have inhalation typically have the highest levels, followed by injection, followed by GA, gastrointestinal, and followed by cutaneous, are fairly low levels. And so one thing that really is interesting is this also corresponds to what percentages survival you have, both with and without treatment. So in summary, LF and PA are the most abundant, but because the detection limits are probably better for LF, it actually makes a better biomarker. EF is quite low, especially in early, but they all do tend to come together at the end. And basically, these are fast sensitive methods that can both diagnose the disease, but also can indicate how effective treatments are going. Um, just real quick, I'd like to thank for the botulism side, Susie Cab and their botulism laboratory, or the toxin laboratory. Also, Carolina Luquez and the National Botulism Laboratory, along with a host of other collaborators, especially Jim Marks that made our antibodies. And on the anthrax, I'd like to thank Dr. Ann Boyer, who heads up our anthrax toxin team, along with Maribel Hanato, Maria, and Adrian Wolfett, Susan Kuklinick that helped develop some of the methods, and great collaborators that we have with Conrad Quinn and Alex Hoffmaster, and a host of people with different clinical investigations. So I'm able to take questions.
We would like to thank our speaker, Dr. John Barr, for presenting today on the interface of biology and chemistry, how advanced analytical techniques benefit the diagnosis, assessment of treatment efficacy, and prevention of toxin-mediated diseases. We will now respond to questions sent in by you, our participants. If you have any questions for our speaker, please type them in the chat area now. From Grace, how cost-effective are these investigations testing?
You know, they actually are quite cost effective, because I know that the equipment is expensive initially, but mass spectrometers actually do a wide range of things. We never have one just dedicated to one test and waiting. These things are used over and over and over, and so once the initial investment is made, really the tester, $40 or $50 a piece for anthrax, and they're even less for the botulism. As I mentioned, I think both of these methods are going to be hopefully moving out into state public health laboratories, particularly because the many of them are investing or haven't already invested in these platforms where where you can put basically a whole organism onto a multi-slide and you get a fingerprint, and so it's a bacterial ID that's very rapid and very cost effective.
Thank you, John. Any other questions from our audience? Well, it looks like we have no further questions. Thank you again, Dr. Barr, for your wonderful presentation. If you would like to receive continuing education credits for this webinar and have not registered, you must do the following. Create an account in Train if you currently do not have one. Email your name and email address associated with your Train account to labtraining@cdc.gov. You will be manually registered and sent instructions on how to assess the evaluation and receive your certificate. As we close our webinar today, please listen to the following information regarding obtaining CEUs, the future archive webinar for this presentation, and additional training opportunities. Thank you for participating in today's webinar. Have a positive and healthy afternoon.